Hi, my name is Haley Runberg, and the research article I chose to discuss is titled Long-Term Dietary Patterns Are Associated with Pro-Inflammatory and Anti-Inflammatory Features of the Gut Microbiome. This paper is by Bolt, Vick, and Iman, and it was featured in Gut in 2021. So their research highlights the importance of diet on inflammatory disease states and the prevalence of certain bacteria depending on the diet. I chose this paper because I'm very interested in gut microbiota from the genus and species level and focusing on which bacteria predominate, their functions and their benefits, and how you can increase or decrease them based on diet. I think as more interest and research goes into the gut microbiome, the function of each gut anaerobe will be better understood and how to increase the most important ones, especially as in this research in preventing inflammatory diseases, which we will discuss in detail. So irritable bowel syndrome was included in the study, though in studying biopsy tissue, there really is inconclusive information on whether there is in fact true inflammation happening. It may not be classified as a true inflammatory disease, just as an irritation of the large intestine. But ulcerative colitis and Crohn's disease are both classified as inflammatory diseases. So in ulcerative colitis, the inflammation is happening mostly on the inner lining of the colon. And in Crohn's disease, it can happen anywhere along the digestive system, and it affects all layers of the bowel wall. So the image on the right shows what's happening in inflamed mucosa versus healthy mucosa. The healthy intestinal tissue has epithelial cells that are protected by an intact mucus layer, as you can see in the yellow. This allows a barrier between the intestinal cells, between the interstitial space and bacteria. The inflamed intestinal tissue is compromised with a decreased mucus layer, and it causes a bacterial influx through the epithelial cells. This leads to leukocyte infiltration and inflammation, as well as a potential for autoimmune reactions to occur. So first I want to show you guys these maps, which I thought were really interesting. Ulcerative colitis is here on the left and Crohn's disease is on the right. The maps show hardly any cases before 1960. It started to increase from 1960 to 1979, and then you see a rapid increase from 1980 to 2008 when this was published. We know through copious research that the Western diet has a lot to do with this, but it was a little shocking to me to see how Canada and Alaska are really as high as the US, and then parts of Europe as well. So this is a more recent map of all inflammatory bowel disease occurrence in 2021. You can see here that Africa is starting to see an emergence. Asia, parts of Europe, and South America have an acceleration of incidence. And Australia, other parts of Europe, and North America all have a compounding prevalence. So this map really represents how there's a major issue, and we really need to put our collective efforts into more research and figuring out what's going on and why there's a worldwide prevalence of IBD. So why do we think this happened? Like, what could some of the causes be? Well, over here in the second graph, we see how industrialization has a lot to do with the rise. And when it first happened, there was a shift in economies from rural to urban, so from agricultural to manufacturing after the Industrial Revolution. And the population density in cities led to a change in diet, which we now refer to as the Western diet. Another major component that we can't leave out is genetic. Um, so inflammatory bowel disease has been linked with mutations or dysregulation of mucin family genes, glycosylation genes, autophagy genes, and inflammatory genes. Certain genetic abnormalities can impair these processes, which can disrupt the gut lining. Here in mucin family genes, as in the name, they play a major role in providing an intact mucus layer. With glycosylation genes, epithelial glycans provide nutrients for commensal bacteria, and they play a role in regulating gut microbiota. And then in autophagy genes, they're involved in cell survival due to cellular component recycling and homeostasis. Inflammatory genes are involved in immune response and inflammation. So any disruption of these genes could lead to IBD in varying levels of symptoms and the type. Crohn's disease has a high incidence of autophagy, mucin and inflammatory gene-related genetic mutations, whereas ulcerative colitis involves mucin and inflammatory gene mutations. And then we have irritable bowel syndrome, which has no genetic markers that we know of, but it is potentially caused by diet, stress, and lifestyle. So in this research paper, they focus solely on diet's relationship to inflammatory bowel disease. So that's what we will focus on. And here's the bottom line up front. Diet affects specific bacteria that may either promote or diminish gut health. So this study shows that inflammatory diseases, Crohn's disease, ulcerative colitis, and irritable bowel syndrome have a strong correlation to certain foods and proliferation of certain bacteria due to diet as compared to healthy controls. Diet content, quantity, and timing all shape gut microbial composition and function. The next point is that gut microbiota modulation has the potential to prevent inflammatory diseases. So long-term diets with high amounts of vegetables, fruits and nuts, legumes, fermented dairy, and fish, and a higher intake of plant versus animal foods, while avoiding high amounts of alcohol, processed foods, and soft drinks could potentially prevent inflammatory bowel disease. And the third point is further analysis of gut microbiota and their function is important for future dietary interventions. So with dietary modulation, we could see an increase in more short-chain fatty acid synthesizing bacteria and bacteria that have known anti-inflammatory effects versus those that are causing the health conditions that keep rising. Inflammatory bowel disease is a disease that affects the homeostasis between gut microbiota and the intestinal immune system. And so there are actually a growing number of immune-mediated inflammatory diseases that are being linked to gut microbiota dysbiosis. And some of these are arthritis, diabetes, autoimmune diseases, and even depression. So because it's so far-reaching, it makes it that much more important for research to be poured into this topic. Okay, so let's get into the study. 1,425 individuals were tested. 205 had Crohn's, 126 with ulcerative colitis, 223 with irritable bowel syndrome, and there were 871 healthy controls. So these inflammatory diseases were actually diagnosed by their physician using histopathological, radiological, and endoscopic methods. One stool sample was collected and then immediately frozen, picked up, transported on dry ice, and then stored at -80°C. Inflammatory markers were measured using the same sample, which I think is really good. It makes it more reliable when it's coming from one sample instead of multiple. So that was good. DNA extraction was performed using All-Prep DNA RNA kit. Shotgun metagenomic sequencing was done using Illumina HiSeq, which we know is great for bacterial genome sequencing and is great for large amounts of data. And a need data toolkit was used to trim reads, trim read results for quality control. Reads aligning to the human genome were removed using need data integrated Bowtie 2. And the quality of processed metagenomes was examined using the FastQC toolkit to analyze raw sequence data that could affect downstream analysis. Functional profiles were calculated using HumanN2. And along with that, taxonomic composition was evaluated using metagenomic phylogenetic analysis. Microbes and functions found in less than 10% of samples were omitted from the analysis. And a dietary intake assessment by the Food Frequency Questionnaire was collected with each fecal sample. So this questionnaire assessed how often a food item was consumed over the previous month on a scale and then along with the amount. So this is an example of what they used. Okay, this table contains information about each cohort and many characteristics that may play a role in the outcome of the study. Some of the key findings in this table are those with Crohn's disease and irritable bowel syndrome were typically younger than the healthy controls and those with ulcerative colitis. More women were diagnosed with irritable bowel syndrome, and more men had both Crohn's disease and ulcerative colitis. There were more smokers in other cohorts as compared to those with ulcerative colitis. Medication usage was accounted for in this study with no real impact on the results obtained, so that was good. The bottom part of the table is all the main food groups in grams per day, which we will discuss. This is a cladogram of all of the common food patterns and pairings that were identified, so the clustering. Some examples are cereals were clustered with dairy, meat was clustered with potatoes and gravy, and a fast food cluster consisted of meat, French fries, mayonnaise, savory snacks, and soft drinks. There were 25 patterns that resulted from the food questionnaire. We'll go over most of the important findings in separate slides for each food group and the resulting bacteria using this data. Okay, here we see dietary groups involved in metabolic pathways using a forest plot. The x-axis represents the association coefficient, and the more significant the association, the larger the dot size. So the most important findings here are that the cluster of fish and nuts and bread and legumes have a negative association with several pro-inflammatory pathways, such as those that are predictive of E. coli. The bread, legumes, and fish and nuts group is involved in anti-inflammatory pathways, such as fermentation to acetate, creating short-chain fatty acids, biotin synthesis, and detoxification of ammonium. And then I didn't want to leave this one out. Dairy and lean beef was involved in L-glutamine synthesis, which also has anti-inflammatory effects. Then on the right here, you see a fecal calprotectin test was done. It was done on each sample of patients with Crohn's disease and ulcerative colitis. And it's commonly used to test for GI inflammation. So calprotectin is a protein in your stool that's commonly found in small amounts, but in large amounts, it's an indicator of irritation and swelling in the intestines. This test is great because it's quick, inexpensive, and non-invasive. And so the results were that a fast food and a high meat diet increased fecal calprotectin, while a diet rich in fish and nuts resulted in low calprotectin. Okay, there was a positive association with meat consumption and total fat intake with microbes that are found in the oral cavity and upper GI tract. The thought is that microbial carbohydrate fermentation creates an environment that is mildly acidic and will inhibit the growth of these bacteria. So switching to a high-fat diet can affect the colonic pH and then allow for those that are found in the upper GI tract to grow in the lower GI tract. So in the lower GI tract, there are true anaerobes that thrive and make up most of the bacterial profile in your gut. And as the environment becomes more oxygenated, facultative anaerobes can thrive, and many of those have inflammatory effects on your gut, which is shown here on the left. So as you can see here, the bacterial load decreases with higher oxygen and lower pH, and the mucus layer is less thick. Then the opposite is true. With less oxygen, the pH and bacterial load increases and the mucus layer thickens, as happens in the lower GI. So this chart on the right shows that the homeostasis of decreased oxygenation allows for true anaerobes like Faecalibacteria to thrive. But with increased oxygen, which lowers the pH, it can lead to facultative anaerobes like Enterobacteriaceae to take over, which results in the term dysanaerobiosis, which I love, rather than just dysbiosis. Okay, now we're going to get into which bacteria were found in high numbers in the gut when eating certain foods. So this is the fun part. Okay, and again, I think that this is so important to try to figure out which bacteria are predominant in certain disease and health states, which I think this paper did a good job of identifying ones that increased due to diet and the patterns that they saw. One of the most important things that I think we could ever learn is how these microbes interact with each other and how they function to protect our gut. So this research focused on each bacteria and whether they induce an inflammatory response or not. And of course, for the sake of time, like we can't get into the all of the known functions of these microbes, but just which diets increased which bacteria, and if they do things like synthesize short-chain fatty acids that are known to reduce inflammation, or if certain bacteria have known inflammatory effects, and simply which commensals they found increased due to a certain diet. So across all cohorts, even healthy controls, Faecalibacterium prausnitzii and Roseburia hominis were predominant in a diet that was rich in nuts, fish, vegetables, and cereals. Of course, these bacteria are both known for their anti-inflammatory effects. Bifidobacterium was found in high amounts in diets high in bread and legumes, with many acetate synthesizing bacteria, and had lower numbers of E. coli, Bacteroides fragilis, and Parabacteroides. Fruit and red wine, or any foods with high levels of polyphenols, had high numbers of Faecalibacterium prausnitzii, Eubacterium hallei, Ruminococcus obium, Anerostipes hadris, Ruminococcus lactaris, Allostipe species, and many that produce acetate and butyrate. However, with this group, there was a lower abundance of Bifidobacterium species, which was actually a little surprising to me. But unsurprisingly, the plant protein group had a very high level of Bifidobacterium species. And of course, that's known for anti-inflammatory effects. Fermented dairy, unsurprisingly, led to a high amount of lactic acid bacteria and fermentation to butanediol and peptidoglycan synthesis. And this is a really cool one. Coffee drinkers had a high amount of Ocelobacter species, which I'm largely unfamiliar with, but I am a coffee drinker, so I need to know this one. And also various glycolytic pathways and heterolactic fermentation. So a diet high in fast food showed higher amounts of Blauschia, Lactobacillus bacterium, Clostridium bolte, Coprobacillus species, and Ruminococcus. Fast food and savory snacks showed a high amount of Parabacteroides johnsonii, Lactobacillus sakii, Lactobacillus, and Ruminococcus species. Processed food showed Proteobacteria, Akkermansia muciniphila, and Ruminococcus navis with increased gut permeability. High sugar and pure grain alcohol showed a higher abundance of quinone synthesis pathways that were also shown in IBD. And an animal protein diet other than fish showed high amounts of firmicutes, Erysipelotrichaceae, Blauschia, Ruminococcus, Streptococcus, with low bifidobacterium, except for Bifidobacterium dentium, which is typically found in the upper GI tract. So this correlates back to the previous chart about how animal protein has the ability to possibly lower pH and allow for the upper GI tract organisms to grow. As previously mentioned, Faecalibacterium and Rosburgia are both capable of short-chain fatty acid production, and they have anti-inflammatory effects, providing protection of intestinal mucosa by fermentation of pectins and fiber to butyrate and acetate. So these are two bacteria that in high numbers are markers of good gut homeostasis. In this figure, red is a positive association and blue is a negative association. It shows that Faecalibacterium is positively associated with consumption of fish, fruits, red wine, and lean beef, while Rosburgia was positively associated with fish, nuts, legumes, vegetables, cereals, and plant protein, but has a negative association with soft drinks, high sugar foods, and high amounts of simple carbs. And this was observed across all cohorts. Also in let's see, Rosburgia's chart, you see that processed cheese had a negative association, and there was a negative association with applesauce with Faecalibacterium. So maybe that is from the high sugar content. These were the common denominator diets and the unique bacterial profiles found with each inflammatory disease. In Crohn's, irritable bowel syndrome, and ulcerative colitis, bile-tolerant bacteria were in high numbers. Bilophila, Bacteroides, non-butyrate-producing allostype species, and Sutterella, they were all associated with fast food and ready meals. In Crohn's disease, there was an abundance of Bacteroides vulgatus. That's associated with milk, animal protein, fat intake, and soft drinks with a lower vegetable and protein intake. And in irritable bowel syndrome, low Doria species was seen and high Bifidobacterium species. They had a diet high in bread and cereals, though a lower caloric and total protein and plant intake. So do you guys think that the fact that irritable bowel syndrome isn't known as a true inflammatory disease could be that Bifidobacterium helps protect from inflammation, potentially? And then in ulcerative colitis, Methanobacter smithii stood out, which is a methane-producing bacteria, and an association of a diet high in milk, sauces, butter, sweets, and alcoholic drinks were made. So the key takeaways here are further analysis of gut microbiota can lead to understanding their function, which bacteria are more important in maintaining gut health and IBD prevention. Animal-derived foods, sugar, alcohol, and processed foods resulted in an increase in endotoxin synthesis pathways and increased intestinal inflammatory markers. Plant-based foods, fish, nuts, and legumes are associated with advanced pathways of nutrient metabolism and short-chain fatty acid production, and a decrease in intestinal inflammatory markers. And lastly, that diet can alter gut microbiota to help prevent inflammatory disease. So my hope is that with more microbiota research like this, naming exactly which anaerobes increase or decrease with certain foods, and as we learn more about which bacteria directly affect the gut, it'll be useful to recommend certain foods for more of a long-term prevention or intervention as necessary. Some of the limitations I think that should be mentioned are that the study is from a single point, it's cross-sectional, and it cannot identify causality. The food questionnaire is based off of memory from a previous month and may not be completely reliable. Long-term diet is also based off 1 month, which is not a true indicator of a long-term diet, in my opinion. Their diet might have changed over time, definitely longer than a month period. It probably should have been based off of years, but you know, how reliable would that be too? But yeah, just 1 month didn't seem very long to me to call it long-term. So some questions I have for you guys are, do you think that genetics play as much of a role in inflammatory bowel disease as diet? Or which do you think has a larger impact? Where do you think the future is with probiotics or fecal transplants and helping with gut dysbiosis? Do you believe that there's a window of time to colonize the gut, or that probiotics can help enough to actually restore the gut? Do you think that the map of IBD will keep growing or decrease with more knowledge and research? How do you think industrialized societies can combat routinely eating a Western diet? Because Western diet equals convenience. So I don't know, that's a hard one. Do you think schools should implement healthier eating to create healthy habits early? If this, you know, truly is a long-term diet issue, you know, starting healthy eating early would be very important. Okay, so thank you guys for listening to my overview of this report, and here are my references.